Senhoras e senhores, bom dia. Solicitamos aos presentes que ocupem os seus lugares. E para que o evento transcorra com tranquilidade, pedimos que os celulares sejam desligados ou colocados no modo silencioso. Tem início a solenidade de abertura do evento, a Neurociência do espaço e do tempo, das células individuais às populações neurais, com o Prêmio Nobel de Medicina Edvard Moser. É com grande satisfação que a Universidade Federal de São Paulo recebe o Nobel Prize Inspiration Initiative, programa da Nobel Prize Initiative e AstraZeneca, que tem como objetivo envolver e inspirar jovens pesquisadores. A iniciativa propicia a disseminação de diversas descobertas científicas no mundo ao levar premiados Nobel a universidades e centros de pesquisa de diferentes países. Neste momento, convidamos a reitora da Universidade, da Unifesp, professora Rayane, para que se assente à mesa. Está faltando uma cadeira. Chamamos agora a professora Susan Pantarotto, pró-reitora adjunta de pós-graduação e pesquisa, para que se assente à mesa. Chamamos agora a diretora executiva de assuntos corporativos da AstraZeneca Brasil, a senhora Marília Guzmão. Então, para dar início aos pronunciamentos, convidamos a diretora executiva de assuntos corporativos da AstraZeneca, a senhora Marília Guzmão, para o seu pronunciamento.
Obrigada. Olá, bom dia. Primeiramente, gostaria de agradecer por nos receber aqui na casa de vocês. É uma grande honra estarmos acompanhando esse segundo dia do laureado do Prêmio Nobel no Brasil. Em nome da AstraZeneca, eu tenho algumas palavras. Gostaria de agradecer a presença de todos e todas. É uma grande honra estar aqui para celebrar um momento especial para o nosso país, a vinda de um laureado do Prêmio Nobel, e a presença desse cientista junto à comunidade acadêmica, profissionais de saúde, e todos esses dias que a gente tem feito esse circuito, né, apresentando, introduzindo e conectando a ciência com a sociedade civil, tem sido momentos de grandes aprendizados, não só para nós da AstraZeneca, que vivemos de ciência, né, e promulgamos a ciência, mas também acho que para todo o ecossistema que trabalha e acredita na saúde. A AstraZeneca é uma empresa que está há aproximadamente 25 anos no Brasil e que já há alguns anos tem esse compromisso de trazer laureados e cientistas comprometidos com a ciência e a saúde para essas conexões. Então, esse evento, ele acontece já há alguns anos e o propósito, ele sempre se renova. A liderança da AstraZeneca no Brasil se expressa não apenas por trazer esses cientistas para dialogar com o país, mas pelo conjunto de iniciativas que a empresa promove em pesquisa, inovação e colaboração com instituições brasileiras. Atualmente, realizamos mais de 130 estudos clínicos no Brasil, promovendo saúde, ciência e acesso. Por isso, a chegada de um Nobel ao Brasil, apoiada pela AstraZeneca, representa muito mais do que um encontro acadêmico. É um gesto de confiança no potencial brasileiro, um estímulo à nossa comunidade científica e um convite para que ampliemos a nossa ambição. É também uma oportunidade para aproximar a indústria, a sociedade, a academia e o setor público, em prol de pacientes, pessoas, sociedade e planeta. Que possamos aproveitar o dia de hoje, abrir novas portas, iniciar novas parcerias e inspirar novos caminhos. Que as ideias compartilhadas hoje aqui sejam uma referência para transformar projetos, pesquisas, políticas públicas e muito mais. Agradeço profundamente à universidade, agradeço ao laureado, agradeço ao Adam e os parceiros e toda a equipe que puderam viabilizar esse encontro. E espero que todos aproveitem. Um bom dia a todos.
Neste momento, passamos a palavra à professora Susan Pantarotto, pró-reitora adjunta de pós-graduação e pesquisa da Unifesp, para o seu pronunciamento.
Bom dia a todos e todas. É um prazer estar aqui hoje. Em nome da nossa pró-reitoria de pós-graduação e pesquisa, agradeço muito a oportunidade, a parceria, a presença de vocês aqui. Em nome da Daiane, da Thalita, da Bia, em toda a equipe de organização dos eventos da AstraZeneca, agradeço a vocês a empreitada, a parceria, a confiança em estarmos todos aqui juntos hoje. Em nome do Adam, agradeço por nos dar a oportunidade de receber o Dr. Edvar aqui na nossa casa hoje. Agradeço a todos e todas da nossa instituição por estarem aqui conosco. Realmente é muito gostoso, é muito importante para nós vermos o auditório lotado da maneira como se encontra. É realmente estimulante para a nossa pró-reitoria de pós-graduação e pesquisa, quando nós temos um evento e temos esse tipo de público, composto por alunos, por tais, por docentes, enfim, por nossa comunidade Unifesp. Em nome do Michel e da Lidiane, agradeço a STI por todo o apoio aqui à realização do evento. Em nome da Tamires, agradeço a nossa equipe da Pró-Reitoria de Pós-Graduação e Pesquisa, por toda a empreitada que estamos juntas, né? Todas as ideias que nós trazemos, elas vestem a camisa e nós conseguimos realizar. Em nome da professora Rayane, agradeço muito por estar aqui conosco e o voto de confiança. Muito obrigada e um excelente evento a nós todos.
Neste momento, convidamos a reitora da Unifesp, professora Rayane Assunção, para dar as boas-vindas a todos os presentes e proferir algumas palavras.
Bom dia às pessoas presentes aqui, participando deste evento. Então, cumprimento aqui a professora Susan, em nome dela, cumprimento toda a equipe da ProPGPQ, que é a nossa pró-reitoria de pós-graduação e pesquisa, que se colocou nesse contato, né, nessa parceria para poder realizar esse evento. Em nome aqui da senhora Marília Guzmão, agradeço a AstraZeneca por ter reconhecido a nossa universidade como uma instituição que, de fato, tem produzido conhecimento, tem produzido pesquisa e, portanto, seria um lugar que deveria acolher alguém como o pesquisador, o laureado aqui, senhor Edvar Moser. É um prazer, é uma alegria, uma honra poder recebê-lo aqui na nossa instituição e a sua presença é motivo de estímulo para a nossa comunidade. Então, aqui eu vi estudantes de vários campi da nossa universidade. A nossa universidade, né, a Universidade Federal de São Paulo, hoje é uma comunidade composta por 25 mil pessoas, as diversas áreas do conhecimento, mas nós temos uma história, né, de mais de 90 anos de produção do conhecimento na área da ciência da saúde, na medicina, nas ciências básicas, né, que possibilitam a pesquisa clínica e, portanto, né, um processo de produção do conhecimento que tem como objetivo produzir, né, referências para que isso possa ser aplicado enquanto política pública, enquanto assistência à saúde para a população brasileira. Então, essa é uma comunidade que, ao receber o convite, né, de ter aqui presente um laureado, né, que tem colocado a sua carreira à disposição de promover ciência, e ao mesmo tempo de estimular novas pessoas, né, no processo de produção do conhecimento, é algo que muito nos estimulou, algo que muito nos fomentou para fazer com que essa divulgação trouxesse a esse anfiteatro hoje pessoas dos diversos campi da nossa instituição, que são 7 campi espalhados por várias cidades aqui do nosso país, do estado de São Paulo, e aqui eu estou vendo pessoas, né, que vieram aí das diversas áreas da tecnologia, da saúde coletiva, da saúde clínica, da pesquisa básica. Então, é um tema que possibilita que pessoas, né, de diversas áreas possam estar aqui hoje motivadas e estimuladas a pensar o quanto que a ciência, de fato, possibilita, né, para a transformação das pessoas, para a transformação do nosso país e para a dignidade da vida das pessoas. Então, deixo aqui, né, a nossa alegria por poder ter esse reconhecimento da parte de vocês. Eu sei que um laureado passa por poucos lugares, então o fato de ter nos escolhido é motivo de muita honra, mas também é um momento de muito compromisso nosso. Eu entendo que a nossa comunidade aqui está ávida por ouvi-lo, né, por poder, né, beber desta fonte, e com certeza, tanto o conteúdo que vai ser colocado aqui, como a trajetória, né, do senhor Moser, é motivo de estímulo para cada jovem, para cada estudante, para cada pessoa que se coloca nesse desafio que é produzir conhecimento, né? Porque ser cientista é uma batalha diária, né? Porque é uma aposta que nós fazemos em algo que muitas vezes é uma pergunta, é uma questão, é um desafio que está colocado ali e tem investimento, né, de muitas pessoas, de muito tempo, de muito estudo, de trabalho coletivo, para que aquilo, de fato, né, seja transformado em conhecimento. E depois tem um outro desafio, que aí as parcerias são muito importantes, por isso que a gente entendeu que essa relação, né, com a AstraZeneca seria algo importante, que é como é que a gente possibilita que aquele conhecimento que foi produzido, ele possa alcançar a população, que a gente diz, olha, a inovação é isso. Inovação é quando eu consigo transformar aquele conhecimento em algo que vai chegar até as pessoas. Então, essa ação conjunta é algo muito importante. Então, agradeço aqui também ao senhor Adam, né, que está nesse trabalho conjunto, né, de poder trazer o laureado e também fazer com que hoje seja um dia, né, de aprendizagem, mas também de inspiração para a trajetória de tantas pessoas aqui. Então, deixo o meu agradecimento mais uma vez a todas as pessoas que se envolveram nessa organização e todos aqueles e aquelas que vieram aqui hoje beber dessa fonte, se inspirar nessa fonte e também colocar a nossa universidade, porque vocês é que fazem a universidade, né? Então, poder colocar a nossa universidade também em contato, em articulação, em ações conjuntas em prol da ciência, do conhecimento e da saúde da população. Obrigada e bom evento.
Agradecemos, então, a mesa e convidamos as senhoras para que se assentem junto à plateia. Para dar início ao evento, a Neurociência do espaço e do tempo, das células individuais às populações neurais, convidamos ao palco o Sr. Adam Smith, Chief Executive Officer da Nobel Prize Initiative e com o Prêmio Nobel de Medicina, Dr. Edvard Moser. Convidamos as pessoas que estão assistindo online para que enviem as perguntas através do QR Code que está aparecendo aí na tela e que se identifiquem com o nome, o estado e a universidade de onde falam. E aos que estão aqui presentes, após a palestra, também poderão fazer perguntas para o palestrante.
Light. Okay, thank you. Works. Yeah. I think it's all good. Yeah. Can you hear me? The microphone works? Okay. Good. Yep. Start? Okay. So first of all, thank you so much both to the Federal University of Sao Paulo, to AstraZeneca and to Nobel Outreach for making this possible, and also to the audience both here and many places around in Brazil for attending. My task today is to review some of the work we are doing in the lab in Trondheim in Norway about the brain, how the brain works, with a particular focus on our perception of space, location, How do we know where we are? How do we know how to get from one place to another? Our perception of time, durations, what comes first, what comes last, and not the least, how space and time come together in memory. So space, time, and memory will be the topics. My work is focused on the fundamental principles of how the normal brain works, but I will say at the end a few words about how these functions break down in certain diseases, and especially in Alzheimer's disease. So let me then start with our normal human brain. So understanding how the brain works, how it gives rise to our thoughts and feelings, is a huge task. It's very complicated, and it's made difficult by the fact, first of all, that there are so many neurons. So the number of neurons in the human brain is almost 100 billion, 10 to the 11th. And each neuron connects to about 10,000 other neurons through so-called synapses. So the number of connection gets then up to about 10 to the 15th. So the number of connections are huge. These connections is where the information is expressed and stored. So and then taking into account that these connections are very, very small, then finding out what's going on in the brain that gives rise to the products of the brain, namely thinking, feeling, and so on, is a huge task. But I will, this is what neuroscientists do, and I will begin by showing, focusing in on this cartoon of a human brain, or any brain. Consists of neurons, nerve cells. They are electrically active, so that's illustrated by the white signals going through the neurons. The neurons connect from left to right in this case through electrical activity that gives rise to chemicals that go across the so-called synapse to the other side and activate receptors so that the electrical activity is induced in the receiving neuron. So this kind of communication by electrical signals that give rise to chemical signals that again give rise to electrical signals is the fundamentals of how neurons in the brain work. So how can we make a connection from these chemical and electrical events to the functions of the brain, such as sensory perceptions, memory, imagination, imagery, thinking, planning, decision making, language, and so on. This is a huge task. So how can we make a bridge between these levels of understanding? So one way is to zoom in on some functions that are fairly easy to understand. I mean, still very, very complicated, but nonetheless, they're easier than others. And that includes space and time. So finding our way, knowing where we are functions that have been around through evolution from the earliest times because they are so fundamental to our existence. And they are axiomatic in the sense that they are building blocks for all other more complicated cognitive activities, such as, for example, imagination or planning or thinking. So the fundamental nature of space and time was already addressed by early philosophers several hundreds years ago, including Immanuel Kant, who suggested that space is category of mind functions or that decide how we experience the world. So the world is experienced through space. It's not something that we experience from outside, but space is a way of experiencing the world. And through this lecture and through data that we have collected now with modern experiments, we'll see that there is some sense to this general idea. But coming back to how fundamental space and time are, then it's fair to say that this must have evolved very early in evolution. First of all, in human evolution, even the first humans must have been dependent on finding their way and knowing the time in order to survive, both to find food, to escape predators, or to find a mate. All this depends on the skill of finding one's way. And of course, this doesn't only apply to humans. This applies to all mammals and probably all animals. So what you see here is a branch of the evolutionary tree that includes mammals. So you have the common ancestor down to the left, and then it branches off in many directions. You have humans to the top right, you have rodents to the left, you have bats at the bottom, you have monkeys near the humans. All of these are able to find their way and use the same brain structures that are organized in similar ways with the same kinds of cells that operate in the same way. So very much is in common with regard to the mechanisms that mammals use for navigation or for finding their way. It doesn't even only include mammals. It goes way beyond that because, of course, as we know, birds are able to navigate, fish are, and even flies and bees. They're all very, very good at this, and many of the mechanisms are even shared in the simplest insects. So that means we can actually we don't need to study navigation in humans. We can probably find the fundamental mechanisms in much simpler organisms. So we'll begin then with space before I transition to time and memory, which I will spend a little bit less time on. Space is what we know most about, and I will focus mostly on that. And then I want to go back to the early days of psychology, experimental psychology. So in the late 1920s, 30s, 40s, and 50s, there was a psychologist named Edward Tolman who worked at the University of California at Berkeley, and he worked with rats. Rats were navigating in mazes and doing many different things, and he tried to infer the laws of behavior based on observations of those rats. And one of the things that he observed when these rats were walking in mazes, for example, was that they could make shortcuts, they could make detours. And maybe to us that sounds pretty obvious, but it was not obvious at that time, because the common view in this in the behaviorist area, as it was called in psychology, was that we learn by forming sequences between stimuli and responses that occur one after the other. So you have event A, and then comes B, and C, and D, and E. And if you navigate your way through a maze, you link them all together. And then next time you go through the maze, you remember those links, and you repeat it again. But if you make a shortcut and go between two locations in a maze that you have never crossed between before, and the rat or human being knows that this is a faster way to get to the goal, then that suggests that you aren't only connecting places in the maze, you're actually forming an overview or a map of the maze, of the environment. And this is what Tolman proposed, that both we and rats, humans and rats, all mammals, then by implication, form maps of the environment and use those to navigate our way. Tolman called these maps cognitive maps because he thought that they were more than just spatial maps. They were maps that included many things beyond spatial locations, also the events that occur at specific places in the environment. For example, if you sit here and you suddenly meet a friend back at there in the end of the room, that will be part of the map because you will remember it for next time. So this was very controversial when Tolman suggested this in the 40s and 50s, and it remained because it could never be tested in the brain. So at that time there were no methods for finding out if there actually were such maps in the brain. That took a long time to find out, but today we know a bit about the brain's substrates for space, for knowing where we are. And there are some brain structures are particularly important, so I will show two here. It's the hippocampus in red and the entorhinal cortex in blue. So these are in the medial temporal lobe. It's on the side of the brain, one on each side, and they are not on the surface. They're sort of folded under behind, so it's a bit difficult to draw. But this is based mostly on studies in humans, and especially we know that those structures are important for memory, because if these structures are gone in humans due to damage for various reasons, then we lose our memory, and we also lose our ability to know where we are. Getting lost, in other words. So this is what often happens in Alzheimer's disease, because those structures, as I will mention later today, are among the first to be damaged in Alzheimer's disease. But these structures are difficult to study in humans. There are very few humans who actually have electrodes or sensors in the brain implanted, so we can actually listen to the activity of cells in there, and that only happens in very rare cases where you have to implant them, because in some epileptic patients you need to do that in order to prepare for surgery. So much easier to study in rats or mice, and then again the brains are very similar. Just the scale is very different. So you have a human brain at the top, and then you have a rat brain at the bottom. So much smaller, and a few million neurons instead of some 200 million, I think, neurons instead of 100 billion. So the scale is very different, but the organization is quite similar. You see the same. The hippocampus, the red structure, has the same layers, same organizational structures, just folded a little bit differently. And they're located in somewhat different places in the brain, but it's still in the same places compared to all other structures. So they're wired, connected very similarly as in the human brain. So it makes sense to go to a rat brain, for example, and ask, do we see any spatial maps there? And this is what John O'Keefe did in the early 1970s. So John O'Keefe received the Nobel Prize for this work together, which he shared together with me and May-Britt Moser in 2014. And what he discovered was that in the hippocampus there are certain cells that signal the animal's location. So I will show that now by starting a video very soon. So that video shows a rat seen from above that walks around in a box, and it walks around because there are small sprinkles of chocolate, and rats like chocolate, so I will walk and pick up all these pieces of crumbles of chocolate, and if there's chocolate everywhere, it will walk everywhere. So this is just a trick we use in order to get the rat to visit all possible places. At the same time, the activity from one particular cell in the hippocampus is recorded. This is possible because the rat has implanted a small wire, which is about 20 micrometers wide, very, very thin, thinner than the hair, that goes between the cells, and then at the end of the wire, the insulation is removed, and it can then pick up the electrical activity, the voltage changes that are near that location from single cells. So you can record a few cells by doing this. And that sound, that electrical change can then be converted to a sound. So I will now start the movie, and you will be listening to the activity of one cell in the hippocampus while the rat is walking around and finding chocolate. So each time you hear a bop, that is when the cell is electrically active and fires an action potential, as we call it. And at the same time, you see red dots appearing each time there's a bop. And what you can see is that this particular cell is active only when the rat is in the upper left part of the box. Otherwise, the cell is completely silent. So the cell is active in only one box, and for that, in only one place, and for that reason, O'Keefe called this cell a place cell. other cells are active at other places. You see 10 examples in the middle. It's color-coded so that blue color means low activity and red color means high activity. And you can see that those 10 different cells are active in different locations in the box. And that means that if you now imagine you have several hundred thousands of these place cells, and they're all active in different locations, that means that at every possible location in the box, there is a unique combination of active cells, which then means that you can, from that activity, read out where the rat is at any given time. That was the idea. And this was suggested already in 1978 by John O'Keefe to the right and Lynn Nadell, a colleague of him, that proposed that the hippocampus is actually the building blocks of Tolman's cognitive map. So because by the combination of place cells that are active, you can infer where you are in the environment. So the discovery of place cells came as a surprise in many ways, because this is in the middle of the brain, and yet you have cells that are responsive to something very unique, namely the location of the rat. It's almost like a sensory organ that tells where you are. But this is no sensory organ. This is in the middle of the brain. So how can this happen? So you need to understand then what is the network around and what are the mechanisms that create these place cells. And there was no answer to that for many years. But then in the mid-1980s, with a publication in 1990, some progress was made, because another group of people, Jim Rank to the right and Jeff Taubert to the left, discovered that in a neighboring area called the presubiculum, which is shown in the rat brain to the top, there is a yellow area, there's a different type of cell. So what happened here was that Rank wanted to record place cells, and he inserted his electrodes, and they went down into the brain, aiming for the hippocampus. And then they didn't quite hit, but came into this yellow area, presubiculum, instead. And suddenly, that was an area that no one knew anything about. It was totally terra incognita. And but then he found that these cells in that region, they signal something else. They signal the direction of the animal. And if you look at the plot to the left, shows one example cell. The activity of the cell on the y-axis, on the x-axis you have the direction, or the head direction of the animal. And you can see this cell is only active when the rat was looking in a certain direction, regardless of where in the environment, just it was looking in that direction. And in the middle, you see an example of that. So it's a circular box, and anywhere in the box, the cell is active if the rat is pointing its head towards the northeast. So then you have cells that are responsive to location, place cells, and to direction, which are head direction cells. So obviously, if those are relevant for navigation, they must somehow speak together. You need to calibrate your location with the orientation or direction. And that could happen in the third area, the entorhinal cortex, which is the blue area. And we started recording there in the early 2000s. And what you see to the left is again the same box as in the first video. Rat is walking around, picking up chocolate pieces. And now you see a white dot appearing each time a cell from the entorhinal cortex is active. And you can see that, again, the cells are active at certain places and not at other places. But now it's not one place, it's many places. And those clusters of places form a very regular pattern. And I'll show you the pattern here in that plot to the right. So in the background, you have the trace of the animal as the rat was walking around in the box for half an hour. Each of the black dots is where the cell was active. And you can see that those activity locations form a peculiar pattern. It consists of triangles that repeat all over the space. So it's almost like a grid. And for that reason, we call those cells grid cells. It's like a coordinate system. but it's not Cartesian in the sense that it's 90 degrees. It's 3 axes. But in any case, it could potentially be used for measuring distances and directions in the environment. So it's a possible basis for a map, a metric of a map. And then as animals walk around in the environment, you will be in different places in that map. Almost like a GPS in a sense, that based on changes in direction and distance, you will be you can update or monitor the current location. So these grid cells, they came in different variations. So they differed in what we call phase and scale and orientation. Grid cells have a different phase if they're shifted relative to each other, such as the blue and green cell in the top, or they have a different scale, which means that the fields and the distance between them can be different. So here, the blue cell has a bigger scale than the green cell, or they may have a different orientation, which means that they're tilted relative to each other. So this varies between different grid cells. So we found out that these cells are not organized randomly in the environment. They're not just mixed together. They are organized with regard to the scale of the cells. So if you look at a brain map, you have to the left a map, it's a section from a rat brain. The red area is the entorhinal cortex, where the grid cells were recorded. If you now start at the top of that brain area and you go down, you will see that the scale of the grid increases. So it goes from very tight clusters to more faraway parts, and they get further and further away and bigger. So it starts with, if this is the box, then the distance between the clusters is maybe 25 centimeters at the top, and then it goes down to several meters at the bottom. So you have several scales of grid cells. And then you may ask, is this a continuous map that goes gradually from small to large? No, it's not continuous, because if you look at the plot to the right, then on the x-axis you have the position in the brain from top to bottom. On the y-axis you have the spacing or the scale, and each dot is one cell. And you can see that as you go further down, it goes in steps instead of just a smooth curve. So there are 4 steps in this case. When you start at the top, the first one we hit was about 40 centimeters distance, and then it goes to 50, and then 70, and then more than 100, and so on. So we call these different steps modules. So grid cells are, in other words, organized in several sub-maps, at least 4, probably up to 10, that live quite independently, maps at different scales. But that doesn't really tell us very much how you get from one place to other, or even how you know where you are. So we still haven't answered the question of whether there are maps. Because if you want to know if there is a map, then you can't just observe one cell at a time. You need to have many cells and then see if they're organized into maps. So this hasn't been possible. In the early days, we recorded from single cells, one at a time. But now, due to changes in technology, it is possible to measure instead from neural populations. That means hundreds or even thousands of cells at the same time. And that is thanks to the development of a new type of recording electrode that's called neuropixels probes. These are silicon probes shown to the left, so they're simply just chips that you have in the computer, but they are extremely small. So they have 4 wires or shanks that go out at the bottom, and those shanks, there's no longer just a wire like in the old days. It's actually a chip with thousands of spots that can be used to measure voltage changes. So that instead of recording the electrical activity at one place at the bottom of an old-fashioned wire that you just put into the brain, now you have these probes with a with the bottom part going into the brain that have often more than 5,000 recording sites. And that means you can record from many, many cells, thousands of cells at the same time. And an illustration of such a recording is shown here in this so-called raster plot. So this raster plot is a plot where each row is the activity of one cell. In this case, it's just 149 of the more than 2,000 recorded cells. And black means the cell is active, white means it's not active. And they are then the 149 cells are sort of stacked under each other. So this allows us now to look at the combination of activity instead of just one cell at a time. So at any given time, for example, in the red column, you can ask which cells are active, and then you can ask at the next time which cells are active. And the number of combinations of active cells is actually then astronomical. Because if you have 149 cells, like in this recording, and they all behave independently, well, then the only way to plot the activity, the evolution of the activity, is to do it in a 149-dimensional coordinate system. And that's a little bit hard to imagine, but that's what you have to do if they are independent. And what happens now is that while we have solved one problem, the experimental problem, we get a new problem, namely the analysis problem, because the dimensionality here is enormous. So just imagine that on each dimension here, each axis, you have 10 different locations, just to make it simple. Then the number of combinations, if you can be in all those locations, is 10 to the 149th, which is more than the number of atoms in the known universe. So that makes it very hard. And especially if you really want to see the distribution of activity, you can never get to a point where you have sampled all those places. So then new problem. But then we are actually quite lucky because it turns out that the brain doesn't visit all those possible places, not at all. It only visits very few places. And this is because the activity of different neurons is very highly correlated. So some neurons just do the same thing. And it actually turns out that the number of places you can visit in this space is very limited in most cases. That means that you can break it down. You don't need 149 dimensions. You can use classical or more modern dimensionality reduction methods to break it down to, for example, 3 dimensions, and you can still describe the evolution of activity quite accurately. And that makes it more manageable. So if we now do this, we do that. So you start with all the activity of the, in this case, 149 cells, and break down the 149 dimensions to, for example, 3 or even 2. And then you follow the activity on those dimensions, those axes that explain so much of the variance. If you do this on the grid cells and look at the combined activity, this is what you get. So you now get a point cloud, and each dot in this point cloud is one moment of time in the raster plot, like the green one or the blue one. And at different times, the activity then moves around on this point cloud. And it turns out that this cloud has the shape of a torus, or a donut, if you want, or a bagel. It's the same. So that means that the combined activity that these grid cells moves on is actually a periodic structure. It's a torus. And this is the map. This is the brain's map of space. So in that sense, this is the Tolman's map in at least one of them. But it is quite different from what we thought first. And another exciting thing about the movement on this periodic map is that this happens not only when we walk around in an environment, it also happens when rats sleep. When they normal sleep, slow wave sleep, or when they REM sleep, that is similar to dream sleep, the activity continues to walk around on this toroidal or torus-shaped point cloud. So it doesn't stop, it just goes on all the time, regardless of behavior. What behavior does is just to anchor it to places in the environment, but the map is active all the time. But now this has to be quantified, and there are methods for quantifying it. I will not spend time on that, to just show you how it is done. So you can count the number of holes in a point cloud. So this is the collaboration with the math department and people using in the topology division of the math department. What you can do is, in a point cloud like the one with the torus, you can count the number of holes, and that is in any dimensions. If you have n dimensions, you can count the number of holes and quantify the size. In a torus, seen at the bottom, you will have 2 one-dimensional holes. So those are the red rings. They are orthogonal to each other, so different direction. And you have 1 hole, which is the inside of the torus, which is a 2-dimensional hole. And this you can count the size of this hole. So these are equivalent to the length of these bars in the in this so-called bar plot. So you have 0 dimensions. Forget that. You have 1 dimensions. You see there are 2 long lines, so 2 holes, and there's 1 long line in 2 dimensions. So this is for the data recorded when the rats walk in the box, and it suggests then that this is actually what you would expect from a torus. This even happens when a rat is walking in more complicated environments, like when it's walking on a different maze where it has to walk around and find food, or at the bottom when it's sleeping, 2 different types of sleep. You see it's still the 2 long lines in 1 dimension and 1 long line in 2 dimensions, which means it confirms that this point cloud actually is a torus. You can even map the activity of each cell, not onto the environment, as when I played the first video, the place cell or the grid cell, but you can map the activity onto this point cloud, the torus-shaped point cloud, and every cell will then have its location of activity on this point cloud, so that when the rat is walking in an environment or when he's sleeping or whatever, the activity is supposed to go smoothly over the torus, and then different cells get activated, just like it did when the reference was the external environment. But the fact that this happens also, happens independently of behavior and even during sleep, suggests that this is not really a result of sensory inputs coming from outside. This is something the brain does by itself. And this brings me then to the possibility that this map is to a large extent internally generated. It's just something that is there, that the brain makes by itself. And this is consistent with theoretical models that have been around for a long time, which suggests that you get this activity, a bump of activity moving around on the map. So you see 3 different instances here with the yellow color. And this moves around on that map in parallel with the animal's movements in the environment. So when the animal walks this way in the environment, it also walks this way on that map. But the problem, if the map, internal map, was like it's shown here, you will get a problem when it comes to the end of the map, because then questions, will it fall outside the map? And obviously we don't normally fall outside the map in the internal map. We keep track of where we are. And this would be much easier if the map was now circular, periodic. So if in these theoretical models, you actually wrapped the map around at the top to the bottom and from the left to the right, what do you get? You get a torus. So this solves the problem, and the data now suggests that this is probably the way it works, that you have a circular map in the brain, and all you need to do is then to link the brain's map to the environment, but you will never get to an end. So this is again consistent with an internal map that only needs to be connected to the outside world, but is already there from the beginning. But if this is the case and you have an internal map, then the question comes, when do we get it and how do we get it? And that leads then to the question that has been around for a long time, and the empiricist view versus the nativist view. So are maps or any experience or anything in the brain, any ability we have is that a result of experience, or is it a result of internal wiring or innate conditions? So with respect to space, there is a strong innate component, and I will show that with some data that we are currently working on. So this is work of Matteo Guadamagno, who is a postdoc in the lab, and he recorded from very, very young rat pups. So focusing on the green box here, which is the days from postnatal day 0 when they are born, up until postnatal day 15, it goes all the way up to 20. But the important part is the green box, because this is the time when the rats are extremely immature. They have not yet opened their ear canals, so, and the eyes are not open, and they can't walk. They just essentially lie. They cannot walk upright. So at that age, these rat pups are very immature. And Matteo still recorded from them when they were sleeping. So they're sleeping here in this pot. They're sleeping 80 to 90% of their time when they're not feeding. And then he found the following, which you see here. So you see the 3 torus-shaped point clouds there. So even at that age, these cells, there's still more than 1,000 cells recorded. No, actually, sorry, this is about 700 cells recorded in the medial entorhinal cortex, and you see it still has this torus-shaped structure. Of course, you can't select grid cells here because you don't know if they're grid cells, but the way to solve that problem is just that you take all the 700 cells you have, you cross-correlate them, you correlate the activity of each cell against every other cell, and it turns out then that you get clusters of activity that are quite similarly correlated. So they are co-active, they're active at the same time. And those clusters, then if you then take each cluster and ask, do these clusters have torus-like activity, it turns out that a few of them do. So exactly as we would expect if some subsets of the cells actually are grid cells. And this happens, this is just verified by the quantification that I mentioned. So you still see the 2 long lines in 1 dimension and 1 long line in 2 dimensions. And this happens at postnatal day 10, when they're 10 days old. You can see in the plot to the right, when we measure the degree of torus shape, that, and each dot there is one of those clusters. Then across 6 rats in this case, you will see that the appearance only occurs at day 10. So at postnatal days 8 and 9, there is no torus, and suddenly the torus comes alive on day 10. And that brings up then the question, what is it that happens at day 10? And at day 10, there's a major change in the activity of the cortex. So at the top, you see the activity of some hundreds of cells, so time on the x-axis. Each row is 1 cell, and you can see that the cells are active only in what we call bursts. So they're active all together and silent for a long time, and then they're active all together and silent for a long time, and so on. So this is because they respond to inputs from the outside. But then suddenly at day 10, this all changes. So at day 10, the activity of all the cells starts to become very uncorrelated. It's totally independent, and they're active all the time. Day 11 also, you see the same, so they get even more active, but essentially the cortex starts to do its own thing. It decouples from the world, and this happens at the same time as connections start to form inside the cortex. And those connections, including both excitatory and inhibitory connections, are needed in order to form these maps. So that's skip that one. That suggests then that these spatial maps that we have in our brain appear very, very abruptly at about day 10 of life, and they have a strong innate component in the sense that they probably depend on genetic programs much more than anything from the outside. then when they are present, they can be used to align with the spatial world, so that you can create one map for this room, another map for that room, and so on. But that's much easier than actually generating the maps each time you need them. All right, for the remaining time, I will talk a little bit about time and a little bit about memory. So first of all, time. That's much less understood than space. But now that it's possible to record activity from many hundreds to thousands of cells, just like I showed, then it's the work of Benjamin Cantor, who is a postdoc in the lab. Then you can again break it down to this, a few dimensions. And what you then see is what you see here in the plot. So now it's broken down to two explanations that explain most of the activity. In color, you have time, so it goes from white to dark red during the course of 10 minutes. And as time passes, you can see that the activity moves in this space from the left to the right. This is very different from what in an area called lateral entorhinal cortex. In other areas, it just stays in one space all the time, so the activity is stable. It's very different. And this is what we would expect. Activity is kind of stable in the brain, so that you have a stable experience of life. But in this brain area, called the lateral entorhinal cortex, which is the sister area next to the area that has MEC, which has the grid cells, in that area, the activity changes all the time. It always changes, never gets back to what it was. It sort of drifts in the space. And that suggests then that if this activity just drifts and drifts and drifts, that means that you actually, by reading out the change of activity, you can actually tell what the time is. But it is not quite like that after all. So this is not just a simple clock in the brain. And the reason is that this drift is not linear. It doesn't change at the same rate all the time. It changes fast sometimes, and it changes slowly at other times. So I show here 3 examples of tasks. So let us focus now not on the box, but in the middle one. In the middle one, you see a maze. The rat walks up in the middle, and then it gets a reward. And then it walks around and comes back, and then it comes next time it comes to the same place again. There's a reward, there's a chocolate piece or whatever, and it does this over and over again. But the reward is an important event for the rat that is walking in the maze. And then you can see that each time it comes to the reward, if you see at the bottom in the middle, there's a big jump. It's not gradual as in the first place to the left, but there's a jump in the activity when it comes to the important place. So this suggests that the drift of activity that is parallel to the experience of time is actually going fast sometimes, and then goes slow and goes fast. And when it goes fast, it's always when something important happens. And you can see that in the plot to the right too. So this shows the speed of neural activity, how much it changes, and you see at the reward place, then it speeds up. So this is consistent with our experience of time. So first of all, there is a drift in activity that could underlie the change, the perception of time, because it correlates with the change in time. But it is different in the sense that this drift jumps each time you come to what we call a boundary between 2 events. When something is, for example, if you sit in this room and you go out of the room, or if something unexpected happens, let's say a monkey comes walking in here, then that will sort of break the experience into before and after, because it's so significant that it breaks it in 2 parts. And when these important things happen, then there's a jump in the activity in this brain area. But these jumps then, as shown in the bottom here, breaks the stream of activity into discrete or separate events. And these separate events are then stored in memory. And then when you recall the event, you can pull up these separate events separately, and you can reconstruct the experience based on those events, rather than having to call an abstract concept of time that the animals don't really experience. So finally, a few words about memory. So again, I mentioned that the hippocampus in red at the bottom there is shown, is very important for memory. That's been known for more than 75 years actually, when patient HM, now known as Henry Molaison, was operated in the early 1950s. Hippocampus was taken out to treat his very serious epilepsy. And when hippocampus was taken out, then he lost his all daily life memory. He could not remember anything new, really. And since that time, hippocampus has been known to be important for memory. But also, not only memory disappeared, also his sense of where he is. So all new locations were gone. So the question is then, what's the difference between the maps that you have in the hippocampus and the map that I talked so much about on the torus in the entorhinal cortex, and how do they relate to each other? So if we begin with the place cell map in the hippocampus, so again, go back to the first video that I showed. You have different place cells that are active at different places in the box. So here you see 10 different cells that are active at different places. If you now go to a different environment, the relation between those place cells is totally scrambled. So they all go to new locations. There's no relationship between this. They totally randomized or orthogonalized, as we call it. So this is very different from what you see in the entorhinal cortex, where the cells, where you use the same map, the same torus all the time. So this is just verified. So we did experiments about 10 years ago, where we recorded from place cells in 11 different rooms at the same time, or sequentially, one room after the other, many cells at the same time. And we compared the activity in those 11 rooms, so just stacking the cells on top of each other and correlating them. And then you could compare the activity in each room with activity in each other room for the place cells. And that results in the matrix to the right. So each box in that matrix shows a correlation for a given combination of rooms. And you can see that they are almost all blue. That means that there's no correlation. They're totally unrelated. And having so many uncorrelated maps, every map you have for every possible room is, of course, what you need if you want to store memories, because each time you store a new memory, a new place, a new event, you need a new one, right? You can't use an old map. You have to always make it different from what was there before. Otherwise, when you recall it, you will mix it up. So it has to be different. So it's very convenient for memory, and therefore put up this picture of patient HM again. So again, very different from the grid cell map. So the grid cell map is the same map, it's just shifted in XY space, but it's always on the same torus. That then brings up the question, how can you get from one to the other? How can you get from the grid cell map to the left to the place cell map on the right? And we suggested already 20 years ago that that could be done if you had a modular grid cell map. So remember, the grid cells were organized into sub-grid cells or modules, groups that could behave differently. And now, if each of these grid cell maps actually rotated differently or shifted differently when you go from one room to the other, you can actually get many, many combinations. So it could be a way that if different grid cells maps rotate or shift differently, and they all then project to place cells in the hippocampus, you'll get new combinations of a large number. And this is what we tested now with a postdoc, Kristi Lykken, who has recorded thousands of cells from each of the same areas at the same time, and then compared the grid cells by testing all possible shifts and translations or or rotations of the grid cell modules and see which one fits best with the observed result. And what you see is the result of this this analysis. So this is quite a lot simplified, but it shows that for 3 different modules of grid cells, they shift in different directions. So the first one when you go from one room to the other, shifts to the right, next one shifts to the left, and the third one shifts down to the right again, different ways. But it confirms then the idea that you have a rigid grid cell map that is, because it's modular and each module can do its own thing, then when those modules come together in the place cells at the next stage, you can actually create a memory system. So this is how you can get from a rigid spatial map to a memory system. So just summing up that, if you see now one room, you have the grid cells at the top and place cells at the bottom, and then grid cells go each other way depending on which module, then the result is that you get this total scrambling and a completely new map in the hippocampus. And as I said, this is very important because it creates the possibility for generating new memories. So finally, a few words about how these fundamental observations of the brain can relate to disease, what you know about neural disease. So it's a long way from the normal brain to the diseased brain, but in many cases, especially in these cases that I talked about today, there are some quite obvious links. So try to understand neural diseases. I think that by finding the fundamental principles about how the brain works, you can actually understand the basis for many neurological and psychiatric conditions. And those are, I say many, also because they are so numerous. So here you have a table of from a study in Europe. So Europe has about 500 million people. And out of those, if you list all different neurological and psychiatric conditions, in this case 178 million had sometime in their life been affected by one of these neural diseases. So that is anything from an addiction through anxiety to dementia or epilepsy, migraine, psychosis, Parkinson, and so on. So it's a huge range, but they're all related to the brain. So if you understand the normal brain, the idea is that you can actually finally find a better understanding of diseases, but also then by implication treatments. But in this particular case that I talked about today, it's particularly relevant to Alzheimer's disease. And the reason is that the brain areas I've been talking about, the entorhinal cortex in particular, and also the hippocampus, are the first that are in the cortex that are affected during Alzheimer's disease. This is where, in the cortex, where the brain damage starts to occur. So you can see brain sections here from 3 different patients at the bottom in the middle. To the left, you have a normal brain. In the middle, you have what you call mild cognitive impairment. So this is when the beginning stages of Alzheimer. And to the right, you have an Alzheimer brain. And you can see how the density of cells is reduced in the entorhinal cortex. And to the right, you see brain scans from people who only later got Alzheimer's disease. And the red area is in the lateral entorhinal cortex, where I showed the mechanisms for timing, and also the medial is shown where you have the grid cells and all that, and hippocampus is next to it. So it begins in the lateral entorhinal cortex, and then there's a reduced activity. The color shows the reduced activity, and then it spreads, and it goes finally to the hippocampus. So this explains how you get how spatial disorientation and loss of memory are among the first symptoms in Alzheimer's disease. So there is no good treatment for Alzheimer's disease yet, although many groups are working on it, and also AstraZeneca. But the good news here is that this development occurs very, very slowly. It actually takes place over decades. So it's a gradual decline in the activity of the entorhinal cortex and its functions. And it doesn't really break out in visible symptoms really until quite late. So that means the window for interfering once a treatment is available is quite long. So it gives quite a lot of opportunities. So solving that puzzle is important because Alzheimer's is age dependent. You see the white plot in the middle shows how the percentage of population affected by Alzheimer increases substantially from age 65 to 85 plus, when above 85 plus it's more than 25%. And to the right, you see the life expectancy, how that increases. As we all know, population gets older, and in this interval, this is for over a period of about 15 years, the life expectancy is all over the world has increased by something like 3-4 years. So the population gets older. That also means more get Alzheimer's disease, and if there is no cure, the number will in the world by 2015 will change from from 60, 70, 80 now to 150 million. So it's a very important task to solve. So, but while this is an important task, it rests on discoveries and our understanding of how the brain works, how the normal brain works. If you know how the normal brain works, you can also understand what goes wrong in the brain and what leads to Alzheimer's disease. So I hope I've given an illustration of what we have been doing. Some of it's obviously very complicated because of the change or the nature of neuroscience getting much more quantitative. On the other hand, I hope that I have communicated that there is working going on in understanding fundamental principles of brain function, such as how we know where we are in particular, but also to some extent our sense of timing and the mechanisms of memory are being discovered. And the work from the lab that I represent is, of course, totally not my own. It's a lab that I run together with May-Britt Moser and these many postdocs and PhD students from all over the world who work there. And I think without them, of course, nothing of this could be presented. So with that, I've reached the end, and I'm happy to take questions in the Q&A subsequent to this talk. Thank you very much for your attention. Okay. Shall I take off that maybe?
Abriremos agora momento para perguntas e respostas para a plateia. E para fazer a moderação desse momento, convidamos o Sr. Adam Smith.
Thank you very much indeed. So hello, I'm Adam Smith. I'm from Nobel Prize Outreach, which is the international outreach arm of the Nobel Foundation based in Sweden. And first of all, I would like to thank Unifesp for so warmly welcoming us here today. It's just been marvelous to be here. It's lovely to see you all. This program we run with AstraZeneca together. We're taking Nobel laureates around the world to meet the next generation of scientists is something we're very proud of. We've been doing it for 15 years. We've visited about 25 countries together over those years, and we're on 5 different continents, but the country we come back to most often is Brazil. And the reason for that is twofold. One is that it's really nice to work with our lovely colleagues at AstraZeneca Brazil. We have a lovely relationship and we enjoy organizing these. But the other is that we meet such fantastic young people when we come to Brazil. It's just wonderful and it's very rewarding. So thank you again for being here. So this is a chance to ask questions of Edvard. I'd like to thank him for his talk. And I'd like to just start by saying that one thing about that talk is you may not even have noticed the Nobel Prize. It was all about the work, nothing about the Nobel Prize. There was a moment in there when he was describing the discovery of grid cells, first observed in 2005 and the subsequent publications, and that was when the Nobel Prize came. It came in 2014. But it's interesting that you didn't really miss a beat. It didn't stop you. You didn't think, oh, well, I've done that now, we can relax. It's that the search went on and you continue to discover things, and then you start recording from populations of cells. So would you like just to comment on that? I mean, this just continuous and ongoing search.
This work that we received the prize for, we received the prize in 2014, and most of the work was done in 2004, 2005, 2006, and also important work around 2012. But that feels like ages ago, because it was a different era. It's all correct. I don't think there's any mistake, but it is a simple way of describing the brain, where you describe what all the brains do one at a time, which was correct, but it's just a building block. So it is like, if you imagine you're building a Lego tower, right? So I have grandkids now, so I know all about that. But then it is like studying the Lego tower, but just seeing one Lego brick at the same time. That doesn't give you a full understanding of the tower. You have to put them together. And that's what we have been doing over the last 10 years or so, that now the tools are there, both the technology and the mathematical tools that you need to understand how all these cells work together. So it's going from studying the elements, single cells, to studying communities. So it is like in sociology, then you learn very early on that you don't understand societies by only studying one individual at a time. You have to see how they all work together. And that's how we understand societies. And in many ways, that's the same with the brain, that if you want to understand brain functions, like almost any brain function really, including even breathing or heart rate, simplest of the simplest, then you need to see how cells work together. And that's where we are now. So it feels like a different area. And it's certainly going forward at a pace that is at least as fast as it was before.
Thank you very much indeed. Okay, so I really would love to hear questions from the audience here and also from our online audience. And of course, you can submit your questions via this QR code behind me. I know we have some questions. So do we have microphones? One there. And do you have a microphone, Talita? Just that one. Okay. So Talita, let me start. Do you have a question to ask from someone in the online audience, please?
I think we have around like 200 people joining from YouTube. So first question is from Vinicius Carvalho. He's based in the Federal University of Minas Gerais. Actually, it's 3 questions in one, perhaps. What about extending these findings to more abstract cognitive maps, like in the Stretch Bird's task from Tim Barron's group? And are these maps based on spatial maps or built from the completely different principles? And less, even if they involve more dimensions beyond 2D?
Yeah, I agree. It's 2 questions, but thanks for that question. This is obviously a question from someone who knows a lot. So congratulations. This is a very important question, whether the spatial maps that we observe especially in rodents, in rats and mice, whether they actually are maps that can be used for much more advanced functions. And there's a quite common agreement that this is what's going on, that in humans, in primates, that you can use these maps to for abstract spaces, like for example, imagine a network of all your friends, right? Hundreds, thousands of friends. They all have their place in some sort of complex network. You can put them at different locations, and then you can move in this network just by thought. And there is quite indirect evidence, or at least there are ideas from the human literature, and including Tim Barron's, as you mentioned, that there is such navigation. And that it essentially uses the grid cells. So grid cells are used not only for spatial navigation to find a way, which is probably what it evolved for. But then once it had evolved, it could be used also for other kinds of navigation, such as in cognitive space, complex abstract spaces. So the evidence is still somewhat indirect, but it is coming and it's an active area. But the problem is, of course, that then you need to record from the human brain. Now, there are not many such recordings available, but there are a few, and it is coming along. So a very interesting question. The other one also was about the number of dimensions. So what we have studied is essentially either 1 or 2 dimensions. That means flat environments. Of course, you can study 3 dimensions. It still seems from very recent unpublished data that the same torus-shaped structure is present there. If you go to 4 or 5 or n dimensions, then we are not in physical space anymore. Then we are in abstract spaces, and what then happens, I don't know. So I prefer to stop at 3.
Thank you very much for that wonderful question. And it was a very informed question, which is fantastic. But you don't have to ask informed questions. You can also ask very general questions. Does anybody in the audience have a question that they'd like to ask? Yes, one back here. Yes, please. And then we'll come back here. We've got another microphone.
Hi, Dr. Moser. I was wondering, since you touched upon REM sleep and the grid cells and dementia, such as Alzheimer's, I was wondering, since there's a neurodegenerative disorder that affects REM sleep behavior disorder that is highly predictive of Parkinson's disease, if you think that maybe this disorder could be related to it could impact the grid cells and the entorhinal cortex, if it could maybe link to Parkinson's and its spatial disabilities and sort of that, and if we could investigate it somehow, this relationship.
I think my take on it is that grid cell activity is expressed in different kinds of sleep. But if sleep is disrupted, or like in sleep disorders, for example, I think the grid cell activity will most likely still go on quite much as it does in the normal state. So it's expressed in sleep, and sleep is also important for maybe not for grid cells as such, but for the memory downstream in the place cells, because that memory has to be stored in the long term, and then sleep is important for that storage. So it happens offline afterwards. You replay the activity over and over again during sleep, and then the memories get much stronger, and they are also stored elsewhere. That's the common idea. So if you have a sleep disorder, either it's slow wave sleep or REM sleep, then I would imagine that memory storage is impacted. But the grid cells just do that thing almost whatsoever.
Thank you very much. Question here, please.
Thank you for your wonderful talk.
Where are you?
Just here.
Here.
Oh, sorry. Okay, yes. Yeah.
I'm Luciane Covolan from Neurophysiology discipline. I have a question. If those maps, they may change upon different emotional situations, like because you're just measuring one animal at a time, but if you have interaction of animals or animals suffering from like anxiety or depression, does the maps change in terms of space and time?
That's also a good question. I think in the same way as I mentioned in the response to sleep, the map is still present, and I would think that the activity still goes around on the torus, for example, but that is on the grid cells. When you come to the place cells, then you have different maps for different conditions, for different rooms, but it could also be different maps, different combinations of place cells for different emotional states. So, for example, in a situation with high stress or anxiety, could be encoded very differently from a relaxed situation. And then when you call up that in memory later, it will be linked to the emotional state, and perhaps even easier to call if you already are in that emotional state. So that level of differentiation does happen for sure, but I would think it happens more in the hippocampal maps than in the grid cell maps, which are much more rigid. And of course, there's not only entorhinal cortex and hippocampus, there's also other areas like the amygdala, which are very important for emotional memories. And then you would have, that also has its own storage, right? So, which is very strongly related to emotional states.
Can I just say, on the emotional states of rats, one thing that is an important characteristic of your lab, which you didn't get a chance to talk about, is the emotional state both of the people who work in the lab, for which you care greatly, and you do a lot to make sure that the people in the lab are happy, and you also do a lot for the animals to make sure they're happy. Do you want to talk about that very briefly?
Yeah, no, we try to. I mean, ideally, we wouldn't like to use animals, right? But in order to study behavior, there is essentially no way around, because we don't know enough about the brain to replace it with a computer. No way. But then the second best is to treat the animals as well as we can. And many of the rats and the mice, they actually have better conditions than most rats and mice in the wild. So we try to give them the best possible conditions, and also to reduce the number to the least possible.
You do that partly because it also gives you better experimental results.
Absolutely. So it's not only for the animals, it's also for ourselves, because if you want to study behavior, you actually need to have collaborative rats. They have to work together with the experimenter to do things, because a rat or a mouse that's not happy will just sit in a corner and that's it. But we need them to walk around, collect food, and do different behaviors in order to get the rewards, go through a maze and so on. And then you need to be friendly with them, and then they're friendly back. So yeah, that's the way. That's a positive emotion, right?
Exactly. Now, I'm afraid you have a very difficult job getting the microphone to everyone, because I've got about 5 questions in the audience. Yes, I know who you are, and there's one here. But first, one from the online audience, trying to balance.
Yeah,
please.
sure. So Marcos Donizete from University of São Paulo. Does gravity influence neurons, or how do neurons behave in zero gravity environments?
Oh, yeah, that's another good question. I'm impressed. Yeah, so it relates to, I mean, we haven't studied gravity as such, but there is quite a lot of work going on the grid cells and place cells in 3-dimensional environments. So not only when they walk on flat surfaces, but also when they walk vertically, and especially mice, they can walk straight up or straight down along the wall if they can grab their feet on it. It turns out that grid cells and place cells are active there as well, but then the 3-dimensional space seems to be cut up in subspaces that still has a plane, and on those subspaces then it's the same toroidal coding. When you switch from one subspace to another, then you just move into a new place on the torus. It seems like that's the preliminary data. I think the level of gravity is the same throughout. So it's more like I would convert the question to ask how do these cells work in 3-dimensional space, and there is quite some work done now.
Thank you very much, Edward. Please.
I should maybe add that much of that work is also done in bats. That's not in our lab, but because bats fly and they still have the same cells.
Thank you. Please, yes, this lady here.
Oh, yeah, okay. Sorry.
I'm Maria Luisa from Universidade Federal de São Paulo. And I had a question. I remember you mentioned the rats don't really experience the subjective feeling of time. But I was wondering if you think that our human subjective feeling of time would be related to this time map on the brain, or much more related to regions related to emotion or something like that.
So whether our subjective experience is related to the spatial maps you have, not directly, I would say indirectly. So I would say that that our subjective experience of time is related to the drifting activity that take place in the lateral and terminal cortex and in the hippocampus and place cells. So those place cells also express space, so they're part of the spatial map at the same time. So in a way, those cells have to do two things. They have to be stable in order to give us a stable sense of being in the same location. At the same time, they also have to change to incorporate time, and that's quite difficult. They have to do contradictory things at the same time, but I can do that. It's almost like imagine the activity goes like in a spiral. You kind of get back to where you were, but on the torus maybe, but then it's also moving in a different dimension in time. So space and time are strongly related, but also our sense of time and the way we break it up in episodes is a separate mechanism as far as I know. I have to say this is very early days because we don't know much about the time yet, but that's how we think about it. Yeah.
Thank you. Then a gentleman in blue here.
Yep.
Hello, Professor. My name is Victor. I'm from the neurology department. And you mentioned during the lecture that those maps appear during development.
Yeah.
So my question is, are these maps inherited, and what do you know about the genetics of grid cells?
Yep. Yeah, also good questions. I wish I could answer them. Well, it is certainly genetic. Data is determined by gene expression patterns because it comes on so suddenly during development. Within 24 hours, these maps suddenly are expressed, probably because given sets of cells connect and maybe because the cells, when they develop, they find there are guidance cues, and suddenly the right cells goes to the right place and you get the right connections. But whether they inherited across generations, then I would say you have to, then we are talking not about the similarities, which I have focused on how the maps are generated in all organisms. But then, of course, we all know there are variations between individuals, and then the differences, whether they are passed on through generations, that is very well possible, but that we don't know anything about. I'm pretty sure that it is dependent on both genetic and epigenetic programs, but still things that we'll have to find out in the future.
Thank you. Please.
Sir Edward, my name is Andrea Ramponi, and so graduation in psychology, and I'm very impressed with research. Currently, I'm looking into developing prostheses with electromyography technology to read the neural muscular signal, and how the GPS for brain could improve the reading of this signal with the creation and more intelligence and function prostheses.
What was the last sentence?
The
last part of it I missed.
How are creating neural prostheses, intelligence, function?
So you're asking whether the knowledge we have about the spatial mapping system or GPS-like mapping system can be, whether a neural prosthesis can be developed to replace dysfunction, for example, during Alzheimer's disease. I think in principle, yes, but in reality at the moment, no. So neural prosthesis have been successful in brain functions, brain areas that are very peripheral. So, for example, in the early sensory systems, so cochlear implants is an example, and also in motor outputs. So if you're going to read out which neurons, if you want to move an arm and you can't move the arm, right, then you can read out from the activity of cells in the motor cortex when and which arm or how you're going to move the arm, right? So you can tell in advance from the cells. So that means that if you can't move the arm, you can actually send signals to the arm or to an artificial limb and then make it move after all. But it's very different when you're in the middle of the brain, and you can't just replace the neurons because you have to talk to the remaining neurons. So it's a much more difficult task. But I'm not saying that it won't happen. It's just not there at the moment.
Thank you very much indeed. Thank you. Lady there, yes, please.
Nice to hear your talk again, because I had the pleasure to listen to you at Falan in 2022. So fascinating data. Thank you so much. So you mentioned, my name is Deborah Sushek. I'm from the Department of Psychobiology here at this university. And you mentioned, you showed that the torus is already present in very, very early in life, much before any type of learning experience. But one sense that it's already there in the pup is smell. So do you think there are connections from the olfactory bulb to the entorhinal cortex that could help the animals locate themselves in their environment?
Yes, you're absolutely right that there are many things that are not present in the youngest animals, right? They can't see, they can't hear, but they do smell right from the first day, from the beginning. And it's very important for them, for example, to localize the mom. I think for the development of the torus, it is not essential, I think, to form the torus, but it's essential for using the torus. Once it's shaped and you have this internal map, of course, you also use olfaction, and especially in rats, that's very important. You could also use visual inputs or auditory inputs, whatever, to align the torus with the place you are. So, because when you go around the torus, you have to also link it to the structure of the particular environment. So let's say it is this room, then you do that in a certain way, you go out, and next time you come back, the linking is the same, so that you use the same map. You don't have to start over again. And for that, you can use olfaction as well. But I don't think the olfaction directly forms the torus. I think that goes probably by internal cues, but there are strong olfactory inputs into the space circuit, and I'm sure they are used for localization once you have a torus, and maybe also for localization before you have a torus. I would say so, but that's not really a spatial map. It's more like the rat pups probably will go towards the odor, and that is a different kind of navigation. It's just going to something, and that's it. And for that, I think that's probably present from the first day. That's a good question.
Thank you. Please, lady.
Hi, I'm Karina Abrão. I'm a professor in the physiology department here in this university. Thank you so much for your talk. And I'm going to switch a little bit the type of question, because in face of all of those beautiful and very nice questions, I would like to know, how do you conduct your lab when you have to decide what your group is going to study next? And I appreciate that even after the Nobel Prize, you are doing like great advances in the science and even taking new technology, new math to analyze the data. And this is very difficult to keep on time. So I want to have your input.
Yeah, you're raising a very fundamental question. It was one that was up in some discussion yesterday too, right?
Yeah.
I think to run a lab and also to mentor and advise young students, one of the most important things is actually the choice of question, because there are so many things you can do, but not all of them are equally important. But of course, choosing the right question is a dynamic process. I mean, you can start, I think you need to start out with a question that you feel yourself, this really matters. This is something I think is important. And that's subjective. Not everyone would agree about that. But I think that decision has to eventually come from inside, because that also gives motivation. But I think one has to ask, what are the things that are missing? and where would it go? What would really change our view if it were true, and so on. And then things happen on the way that maybe it wasn't quite like that, and you have to adjust. So you have to adjust all the time. So I can't, of course, say what are the important questions, but I think everyone has to sit down based on the knowledge they have in the field, and then ask what makes a difference. What usually those questions are often quite simple, not always, but at least in my field, it's like, how do we remember, or why do we not? Why do we recall better after a night's sleep, and things like that, right? That everyone can relate to, and then try to address those questions. Sometimes they may take a long time to address, but I think begin with a big question that we feel are big.
It's such an interesting topic. Just following up on it, I mean, if people were looking at the papers that occasionally appear at the bottom of your slides, the journals are very high profile. Obviously, you pitch your ambition level at a right that you're asking a question that's about as difficult as you can ask, but you can get an answer. So how do you get that right? Because that's such a difficult thing to manage, the pitch.
No, I mean, it's not always right. That's another thing we also discussed yesterday, the tolerance for failures, because if you do that kind of research, you have to accept that sometimes things don't quite go as they should do, right? You make mistakes, and then you have to reverse or take a turn. I think that's a price you have to pay. That's you have to. If you take higher risk, then it also means that sometimes it goes wrong. But then, on the other hand, the rewards are so much bigger if you make a big jump and you actually get it to work. So it's a balance. But I also have to say then that when you take these big risks, and it helps to have a good budget, so which we now have, right? So it's much harder to do that with limited resources. But as we also discussed yesterday, there are things you can do that don't cost so much, and which is still very important, like theoretical neuroscience, computational neuroscience, which you can do quite a lot of thinking. You can analyze data that are stored and all over the world now. All the data have to be uploaded. So you can still have great thoughts and solve important problems, but it is still a safety mechanism to have be able to retain the students for a certain number of years, so that you can complete the study.
Thank you very much. Thank you. Another question from our online audience, please.
Yeah, so Lídia Silveira from the Federal University of Pará in the north of Brazil. She asks, using a similar perspective, but in an opposite direction, could be that individuals with visual and auditive hypersensitivity be safer against this kind of neural deteriorations? Tricky question.
I didn't quite understand the question. Maybe could you repeat it, Adam? Or?
Well, if I got it right, that can you use this knowledge to protect people in some ways who are suffering from from auditory or visual deterioration? Is that right? Yeah. Basic.
Okay. Not directly, I would say, but I think the knowledge we have is generally it's helpful in the long run to understand every kind of disease or degeneration or dysfunction of the brain, because when we know how the brain works, we can also devise treatments in the end against that conditions. But then it's more because of the general principles. I think in the specific case, then it's harder to say. But on the other hand, also the spatial system can be trained, and that may be helpful to a number of other brain systems. So, for example, if you run orientation, you have to navigate, you have to learn how to use your map of space, then you get better at it. So, yeah.
Thank you. Yes, next question up here, please.
Hi, thank you. My name is Dalva Poyaris, and I'm a neurologist and sleep specialist and professor at psychobiology department here at Unifesp. And I wonder, I have two quick questions. One is the inter-individual variability on those spatial and time perception, and according to sex in humans. And the second question, I'm not sure if it is a fair question, but also whether these people with at baseline with lower abilities, spatial and time perception abilities, are at higher risk to develop early dementia symptoms, again in humans.
Okay, yeah, I mean, this is a question that comes up very often. So let's begin then with individual abilities. So obviously, like all other brain functions, people are different. Some are good at navigating, some are not so good. And there's also been the question very often about whether there is differences between males and females. The answer is not yes or no. So with regard to navigation, finding your way, it depends on the exact kind of navigation. When it's navigation of the kind where you walk out in nature and there are mountains and there are rivers, valleys that you relate to, then in that type of navigation, in rats and in humans, often on the average, if you average many people, then males are a little bit better. But if you now go to other kinds of environments where the landmarks are more close, like in a forest, for example, then it is in many studies reversed. So it really depends on the kind of navigation. And I think, but I have to say, it's huge variation within each sex as well. But I think like every other brain functions, there are some where males are a little bit better, some where females are a little bit better. Like, for example, all verbal skills, usually women are much better than men. So, but again, it's the average. So then, of course, the more interesting question that follows from that is how and what are the difference? Why is it like that? And there I can't say so much. So it's related on many studies that are somewhat descriptive, and I think it would be really important to understand why. Whether it really is related to subsequent neurodegeneration, not that I am aware of, really. I don't think so, really. But I think the only thing I can say is that in general, the more you train the brain and the networks that are affected, and especially the hippocampus and entorhinal cortex, where Alzheimer's disease often begins, is that the more robust you are and the longer you can resist the development of Alzheimer.
So we don't have very long left, but I'd like to get in a few more questions. We have one right at the back here, which I'm delighted by. You might have to stand up so we can see you.
I see you.
Hello, Dr. Edward, thank you for the lecture. It's an honor to be here today. I'm from Santos City, a PhD student from Unifesp also. And I found very interesting the lecture, and I would like to know your opinion about the circadian rhythm. Do you think it can modulate those cells, grid cells, place cells, and as along the day, the response during light or dark can alter the plasticity? No. So the therapies, do you believe it can also work better at night or day? I don't know. I was thinking just for example, on Alzheimer's disease, a therapy would be more successful if applied during night or day. I don't know. I was traveling along this.
Yeah.
Thank you.
There are circadian rhythms. They decide all brain functions, right? So, but and including also grid cells, place cells are modulated by rhythms, not directly or not so much by circadian rhythm, but there are some effects, but by faster rhythms. So you have rhythms that are many rhythms in the sub-second range, millisecond range. There are some that go in minutes. There are even some that go in a few hours, and they're all linked to each other. And those rhythms determine very much precisely the timing of when the cells are active. I think by and large the rough general functions of the spatial map that exist all the time, regardless. But the fine scale coordination may be dependent on timing and also on rhythm, including daily or circadian rhythms, but not to the point that I would say that that would make a big difference for any treatment of Alzheimer's. So there's no effective treatment at the moment, at least not one that cures the disease. But anyway, I wouldn't expect that to have a major role. But let's see. I don't know yet. It's too early to tell.
Incidentally, how are your circadian rhythms doing? Given that you're on a 2.5-week tour.
I was first in California, 9 hours difference, and then in Florida was 6, and now it's 4. So I'm gradually making my way back. But certainly it screws up the rhythms, definitely, waking up at the weirdest time. So that's the way it is. It's not easy to change.
You seem to be doing well on it. Okay, another question from online.
Yeah, so Professor Rodrigo Pelicari from Unesp Diadema, he congratulates, really amazing talk, and it's a remarkable work. The question is there any contribution of an entrained suprachiasmatic nucleus, the central pacemaker, the mind's clock, to a successful spatial orientation?
You said a suprachiasmatic nucleus?
Yeah.
Okay, that's related to brain circadian rhythms again. So it's somewhat similar to the other question. Not really. Well, first, circadian rhythms do influence all cognitive functions. So, shifting time zones, for example, is disruptive, and you can get inattentive and unconcentrated. But I wouldn't say specifically on the spatial map, and certainly not training. But having a regular circadian rhythm is important for cognitive functions in general. So in that sense, I would say yes.
Thank you. Thank you very much. Thank you for the question. Okay, we have one here. We really have to draw it to a close. So let's take 2 quick questions, and then you can give 2 answers, and then we better stop. So please, go ahead.
Hi, Dr. Moser. First of all, thank you for your presentation. My name is Julia. I'm from the biophysics department here at Unifesp. And at the beginning of your presentation, you mentioned more complex cognitive functions like imagination. And I was wondering if you could talk a little bit about what we know so far about how the brain employs the grid maps, for example, in these more complex functions, such as, for example, imagining a place by hearing its description, or maybe recalling a path to give directions to someone.
Yeah. Why do you? Okay, answer that, and then we'll get one more question.
Okay. Yeah, I can answer that quite briefly. So what we think is that if you imagine in your head going a certain route, for example, from here through out of the room through the corridor and so on, that sort of corresponds to some change or movement on the internal map too. But as you can imagine, it's hard to study this because we work with rats and mice, and we can't ask them what they think. So that study has to be done in humans. It can be done in humans, but so far we don't quite know. But there's indirect evidence that when rodents, rats sleep and dream, when they dream, because you can see it from the eye movements that they're dreaming, that you actually have movement in the internal map. So it's early days, but my guess would be that this is what's happening.
Thank you. And here we have a question, please. Stand up.
Yeah. Sorry. Can you hear me?
Yes.
Yeah. So I'm Seiji from Unifesp also. Thank you for being here in person. I hope you have a good time in Brazil. So my question is about the toroidal representation. How invariant is that? And if that changes with learning or in different environments, or is it more like static representation?
So whether how invariant it is, whether it changes with learning, and I think it does. So the answer is both. So the map is very rigid and invariant in some ways because the internal structure is just the same. So it's one map that's applied everywhere. But still, it is also true that you have to apply that map in each different setting. So each time you go to a new room, you have to link the map to the surroundings so that it's stable in that room. And that requires learning. It's a very fast learning because when you get into a new room, that happens over a few seconds and then you have it. But it still requires learning, and also you may explore the map more in detail, specific locations and so on, so that you modify the map a little bit, which takes more time. So gradually you can improve the details of the map, but the general map structure, I would say, in the grid map structure is quite invariant. When it comes to the place cells, then it's, as I said, much more totally different for each environment. So you have new maps for every place, really.
Thank you very much indeed. Thank you for all your questions. It's a fascinating, engaging conversation. It could go on and on and on, but I guess people people have other things to do, I guess. At least some need to filter away. So I want to thank you all online and here for your questions and for being here. And I'd like to thank Edvard very much for his talk. Thank you.
Thank you for all your insightful questions, and many questions that really were very shows that at least some people must have understood something. That's very rewarding. Thank you.
Thank you. What was that? We missed that. Do you have open positions in your lab?
Occasionally, every now and then, but you know, then when people want to come from all the world and we restrict it to about 20-30 people, the selection is hard. But when I get that question quite often, actually, what I usually recommend people to do is to start out locally in a good map, a good lab, and get initial training. And when you are more experienced, then apply and see how it goes. But I would also have to say we are not the only good lab. There are many good labs around in the world, right? So it's a place for more than 20 people.
But let me just ask one question. When you are assessing people to come into the lab, is there something you can say that you look for in particular, the way they apply to you, the way they ask you the question of, can I come? What they have to
it doesn't help to just send an email because I get 3 or 4 each day. So I think going through someone is usually a better way. But in any case, I mean, we look for many factors: experience in the field and knowledge of course, neuroscience and statistics and mathematics and coding experience. It's important to tolerate frustrations because you don't find what you want immediately. So it requires some persistence and not the least nice people who can work together is very important. So
thank you very much indeed. And I guess anybody who wanted a photograph, we could do a group photograph in front here. That would be quite nice. Thank you very, very much.
Okay.
Agradecemos então a presença de todos e declaramos por encerrado o evento. Tenham todos um bom dia.